Hello, good afternoon. My name is Suwan Jayasinghe. Firstly, before I start, I'd like to thank Professor Rohini De Silva for inviting me to give this talk. My talk is going to essentially show you the field of research in regenerative medicine. We are trying to reconstruct tissues for the clinic and for many other applications. For example, to build a tissue outside your body with your own cells, for us to test drugs and various other compounds, so as to understand whether there are any side effects and so on, so that once we can test a drug like that on a tissue which is made out of your cells, and then when there are no side effects, we can actually use those drugs on you, knowing that there won't be any side effects. So in this endeavor, I'm going to give you a bit of history into what's the current status quo and who are the main players, what are the techniques out there, and why we do the research we do. So as you know, modern medicine has done wonderful things and had some wonderful discoveries in fundamental research, which has been translated to the clinic. And you can see that from my first slide, that we are living longer, which is a good thing, but this also brings a bad thing about the fact that once you live longer, the human body is only meant to work so much. And beyond that point, what happens is tissues become dysfunctional due to aging and various other issues. So that is where we need regenerative and reconstructive tissue approaches for this application. Obviously, there is a donor pool, and the donor pool at the moment is oversubscribed because of the simple fact that the demand for donor tissues outweighs what is currently available in the donor pool. So although the donor pool exists, the problem with such a approach being there is that transplantation, although very successful over the many years, the patients have to take a fistful of lifelong immunosuppressants. Now, the problem with that is, we go back to what I mentioned before, is that there is side effects from these immunosuppressants. So you can see that there are some very successful stories out here where people have lived a very fulfilling long life over many years after transplants, which have been very successful. But also, as I said, the transplant service is at a breaking point, and this poses many challenges. And this is where reconstruction tissues, or reconstructing a tissue, becomes, if you like, the holy grail. And obviously, because there is a shortage, obviously the donor pool, the tissues that are available in the donor pool are not really at good quality. And you can see here from this slide, there are patients that have been given various transplants that have actually been rejected and caused deaths and various other complications. In fact, in some scenarios, people have had to wait that long that the patients have actually passed away. So these are the problems on depending wholly on the donor pool. But once again, the donor pool being there is to somewhat has been the status quo, where we go and take lifelong immunosuppressants and so on. But as I said, the holy grail is to take the patient's own cells, grow them outside the patient's body in the quantities, in large quantities, and then mix them in permutations and combinations, and make whatever vessel or soft tissue in 3D and have it fully functionalized, meaning with vascularization right throughout the tissue, because if there's no vascularization, the tissue is not going to be functional for long, unless it's going to be in a petri dish, so that's no good. But nonetheless, building a tissue outside the body and then finally transplanting it to function with its microenvironment is the Holy Grail. Now, I told you that I'll be talking a little bit about history. Now, this gentleman here is Alexis Carrel that you see on the left of your screen. Now, he's been working with Charles Lindbergh, the gentleman on the front cover of Times Magazine with Alexis Carrel. And I choose to show this slide because of the simple fact that Alexis Carrel was the first man in the world that actually showed that you can culture cells outside the human body. Funnily enough, there was a news article recently that some of his technicians, due to the fact that they were extremely scared of him, used to fiddle the results by putting live cells into the Petri dishes so that the next day when he came back to work, the cells, it looked like the cells had divided. Any of you who are interested in these articles, you can contact me, Professor Rohini De Silva or Dr. Guthrie will contact me or give you my contact details, and I'm more than happy to share these articles. It's publicly available. Nonetheless, Alexis Carrel was known for the ability to expand cells outside the human body. The next picture that you see is the book that he wrote with Charles Lindbergh. And the reason I wanted to mention Charles Lindbergh is he's the American pilot who crossed the Atlantic. Obviously he was funding Alexis Carrel's work. So that's the front cover of the book. And Alexis Carrel was also awarded the Nobel Prize for this work. Moving on, these two gentlemen that I show you, Joseph Murray and Donald Thompson, they're very important people because of the simple fact that they produced, or they were the first two doctors in the US at Harvard Medical School who did the first renal transplant. They were very successful in doing it, and these are the articles that came through that work, and both of them were awarded the Nobel Prize. Since then, regenerative medicine has taken quite a central stage for understanding that this might be the Holy Grail and the way to go in order to combat the problems that we face with the donor pool. So this article that I show you with one of my very good colleagues who's written Cato Lawrenson tells you the actual story where tissue engineering came, because there are lots of stories and myths who actually started this field of tissue engineering or regenerative biology for that matter. But this article, I point you to this editorial. It actually shows you who did the work first. Nonetheless, this paper was one of the first papers by Weinberg and Bell that showed that you can actually create a functional vessel using collagen and various processing approaches, which was crude at the time, but rudimentary, but nonetheless they showed it, and that was published in 1986 in the journal Science. Please note the dates because that's very important. Subsequently, this article came out in 1990 in the Journal of Dairy Science, which was done by Gertrude. This article, unfortunately, is not cited a lot, although in my opinion, it has done what tissue engineering actually promises to do. This scientist over at California brought all the cells found in the mammary glands and showed that she was able to show that the tissue functions as it was able to create milk. So that is tissue engineering in my opinion. So once again, note the dates and note the two articles. Now they have, in this endeavor to build a tissue outside the human body and transplant it, although that concept does exist and that's the concept that all of us are following, there are lots of other approaches that people are trying desperately to build organs inside animals. Now, something I must also be very clear about, there are lots of people in the world who say that they are going to build an entire organ. I particularly find that very Star Trek or very futuristic, too futuristic for me, unfortunately, because as we, in my opinion, in my humble opinion, when you are born with certain, obviously with all your organs, whether they are functioning or not, as a function of time, as you age, those the tissues and those organs in your body are conditioned to your height, weight, and whatever. So if you go to change the heart of a person who's very thin, as opposed to a person like me, as you can see, I'm not a very thin person, there are going to be obvious problems that one's going to face. So changing the entire organ seems to be too much of a Star Trek kind of dream to me. And therefore, the school of thought that my laboratory follows is that we hope to be able to patch organs, and rather than replace an entire organ. Now, in this endeavor, there are other colleagues of mine in this field that are actually trying to grow human organs in other animals. You can see here the article in Cell that shows that they're trying to build human organs inside pigs. The unfortunate thing about this is that a pig matures much faster than a human, and therefore the organs will not be fully grown as expected to for transplantation. There are various other implications and various other complications that come from this, but nonetheless, although this is still not fully baked, if you like, it's still in its early stages, so we will see how these things evolve. But at present, my opinion about this is that the actual organ does not have the full functionality, if you like, or longevity for it to be actually transplanted into the body. There are various other applications that various other processes, and this is a process that was published by Doris Taylor, which was discussed initially by Stephen Badiak, who's at the McGowan Regenerative Medicine Institute over in the US. And you can see here that she's taken a rat heart and she's teased away all the cells in the heart. Now you can see that the heart has the extreme left image shows with all the blood in the heart, and once she's actually put her chemical cocktail into it, she's teased away the cells, and you're left around with the scaffold of the heart. Unfortunately, by putting taking away the cells, it is now seen that by removing the cells using this chemical cocktail, you're compromising the integrity of the cells, sorry, not the cells, but the scaffold that you're left with. And once you start seeding it with cells, obviously these are adult cells, so they know where to go in the heart. But nonetheless, once the tissue is being fully decellularized, the actual construct has lost its integrity. And I will show you that some of the work that has been carried out with this procedure on human trials have been resulted in patients dying. This is another piece of work where they've done a decellularized tissue and even taken synthetic tissue and actually bridged a vein, a vessel, as you can see on the lungs, on the panel A, that has actually narrowed down. They actually replaced this with spliced it with a thicker piece. Unfortunately, I think this experiment was reasonably successful, but after a while the patient died, and that is because the vascularization and the structure that they actually used to splice that arrowed part where it has narrowed down actually didn't stay fully rigid as it should have. Hence the patient passed away. Similar work has been carried out, which was led by Paolo Maccarini, and although there were 5-year follow-up results, these results were a bit, in English, in England we say it's dodgy, meaning that it didn't quite hold water. But nonetheless, we actually challenged these results, and there was quite a bit of dialogue between us and this group. But later we found out that the work was not as expected, and the work was retracted. So you can follow up on this when you know all the work that I've just shown you. So you can see that although people have been very, very quick to take on these technologies and try to push it very, very quickly into the clinic, that there are huge issues with this. Now, Laura Nicholson, who's a colleague of mine who I know very well in Yale, she's done some work where she's actually decellularizing an actual synthetic vessel and then recellurizing it and then keeping it in a tissue bioreactor for a rather long period of time for the cells to actually migrate fully into the vessel, right through the walls, if you like. Obviously, and you don't have one cell type, you have 3 to 4 or 5 different cell types, and then form a fully functional tissue. That being said, I think the clinical trials, she's doing phase 3 clinical trials now, and I assume that we will hear about the tests if they are successful or not. But nonetheless, this is an approach to have tissues on demand, which is to some extent plausible because you've got blood banks now, and we've had blood banks for so many years. So why not, or tissue banks? So why not have be able to have vessels and various other constructs in there, so that when we need it, we just cut the piece of the length of what we want, and then use it to transplant whatever the issues are. So the process is, as you can see here, this is actually taken from that publication which she published, which I showed you before on the previous slide. So she makes a synthetic tissue, and then she, a synthetic vessel, then a scaffold, and then she seeds cells onto it. And over a function of time, and we are talking about quite a long time here, because the seeding is either whether it be done mechanically or manually, the seeding is not as uniform as one would expect. So there are some issues there. And obviously, before you go to the human, you actually have to take the actual scaffold has got to be fully integrated with cells and functional. So although this schematic is shown here to be, a very easy flow, I don't think in reality it's as easy as this. Nonetheless, it's a concept that we are all working with. So the problem at the moment in this field is that the ability to grow cells in large quantities outside the human body was a problem. We have to some extent grappled with that. We still do grapple with that to some extent, but with the work of Shinya Yamanaka and various others who have been able to show us that you can go from a fibroblast, which is a specialized cell, back to an unspecialized cell, and basically go again to a specialized cell, brings massive possibilities. So basically, if you take a piece of tissue about the size of a postage stamp, for example, decellularize all the cell types in there, and you start culturing them in outside the human body, you might find that 1 or 2 cell types, or maybe even more, don't grow as fast as some. This is where you bring Shinya Yamanaka's technology of IPS, which is induced pluripotent stem cells, where we take the patient's fibroblast cells from the skin rather than from the tissue itself, meaning from the heart or something. So you don't. It's sort of a good thing because it's a non-invasive way as well. So there's less trauma on the patient. So we can take the fibroblast from the patient from the skin, from an arm or something like that, and then make those by exposing it to 4 growth factors. We can get it to go back to an unspecialized cell, which is a stem cell, and then redirect it to become the type of cell that you want, which is not growing, as I said, from the native cells as fast as you want it. Vascularization is tremendously important. I can't stress enough. If vascularization is not there or not present in a tissue, you can build the best tissue in the world. If nutrients are not made available to that tissue, the tissue is going to die. Biomaterials also plays a massive role in this because of the simple fact that we have to form or provide a scaffold for these cells that we want to grow within a tissue, because of the simple fact that if there is no scaffold, the cells will have to work harder and therefore have the possibility of changing their pheno and genotype, which is not a very good thing because it could become cancerous. Obviously, because if the cells have to form their own matrix, if there's no matrix, they have got to work twice as hard as opposed to having a matrix, and they latch onto that matrix and then they start remodeling it. The uses of all of these is that you can actually start building tissues which can be used for therapeutics, where you can start including medical cells and or genes. Obviously, I'm a great believer of in vivo work and not so much in vitro, because of the simple fact that in vivo is more varied nutrients rather than in the case of in vitro because it's nutrient rich. Nonetheless, as I said, we would make tissues in Petri dishes to start testing drugs and so on to understand side effects. So in vitro is also important. And there are various other applications that you can create from this, to discover drugs and screening mechanisms and so on for toxicology studies and all that. So as you can see, the approaches I mentioned there previously are, if you like, are competing technologies. And the approaches that we focused on is the ability to actually deposit living cells with polymer and all the constituents found in native tissues so as to build a tissue in one step rather than having many steps in order to get to that final step. Now, in that process, there are two methods that I categorize them as two methods, direct and indirect. Direct approaches are those approaches that can, if you like, print living cells, a wide range of cells, meaning multicellular populations in large densities with polymer and various other materials like growth factors and cytokines and so on and so forth. Indirect methods are methods that you can use where, for example, you can stamp a structure, like stamping the word confidential on your letterheads, and then the print of confidential is printed with proteins. And then when you expose that to cells, the cells will seed onto the word confidential. The next, obviously, layer by layer, build it up. The direct methods is more of a additive manufacturing 3D printing approach. There are lots of approaches here in the currently that are competing. Like I told you, soft lithography and dip pen lithography are approaches that you use either light or either, if you like, potato stamping. If you like, as children, you might have, I'm sure you'd have cut a potato in half, made it as a stencil, and then dipped it in ink and used it to print on paper. That similar approach, obviously in a much smaller scale, is used. Unfortunately, the thing is, cells, you can't tell cells when to get off and when to get on. So therefore, printing a single layer of cells is fantastic because you can just get them to seed onto the structure that you just created, as, like I said, the confidential word. But putting another layer on top of it is next to impossible, because if you put the printing stamp on top of it again, you will squeeze and kill the cells. Replica molding is a similar technology like that, and has similar issues with that. Now, hanging droplet, I'm sure you have heard of this, where you have use gravity for the cells within a droplet to come together and make a cellular ball. Unfortunately, with that technology, the cellular balls that you create are single balls, or if you make them multi-core structures like a boiled egg, you will find that the inner core structure starts to undergo apoptosis, or central necrosis is what it is called, and that is for the lack of nutrients. So I don't know, they're using various approaches in hanging drop technologies, to build these spheroids, or what do they call them? They refer to them as organoids now. I don't know how they check their viability. In my opinion, I'm a bit skeptical about that. There are other approaches like cell sheeting and organ weaving, which are also available, but unfortunately those as well have various issues with it. So moving down, I've spoken to you about decellularization and recellularization. Lab on a chip is a technology that you build an entire tissue on a lab. Unfortunately, the problem with that is that everything is very, very precise, and it is in at best in 2D and in very, very small quantities. In the direct printing technologies, inkjet printing has done a great deal in this endeavor. There are many manifestations of inkjet printing or 3D printing, but the unfortunate thing is 3D printing uses very fine needles, and that shears cells. And therefore, I will show you some articles that actually show that 3D printing kills cells because of shearing. We actually did some experiments in early 2000, 2001, 2003, and we found that cells were dying. We didn't report it, but to date, people are saying that you can process cells. And there's another community that also publishes about 3D printing of cells, saying that cells are dying. The technologies I'm going to be talking to you about are the last four technologies: bioelectrospraying, cell electrospinning, and aerodynamically assisted approaches. I don't think I will be able to talk to you about the last two, but I certainly will talk to you about bioelectrosprays and cell electrospinning. Both these technologies have now entered the clinic. They have gone through phase 3 trials and are currently being assessed for their for their efficacy and for their final checkpoint before they enter medical hospitals in the US through Pfizer. So in this endeavor, you can see all the approaches in red are the approaches that we've pioneered and discovered in my lab, and I will talk to you about them now. Now, as I said, the problem with 3D printing is that when you build an arm, and for example, if you hold your arm out, it has no supports. 3D printing has to print supports to hold that arm out. That's a waste of material and time. Therefore, I will show you a technology using electrospraying where you don't have to build supports to hold an overhanging structure. In 3D printing, the cell densities that are handleable are very low density. Therefore, building tissues that are human scale relevant are near impossible. The cell types and cell types are obviously different cell types are different size, and therefore they are highly shear. They undergo high shear rates within the needles. Added to that, the cells are not only being sheared, but the hydrogels that you can use, the viscosities are also low. Therefore, once you actually deposit them, they spread rather than maintaining their integrity. Interestingly, cell viability has never been assessed in the way that we assess cell viability. And the reason why we say that we assess cell viability in a very in detailed approach, we've used clinically relevant readouts, meaning flow cytometry, deep sequencing, RNA sequencing, to microarrays, karyotypic studies. None of these studies have been carried out in 3D printing studies. Therefore, we cannot compare our results with those 3D printing results because they have not been phenotyped or genotyped as to the molecular level we have done. Added to that, our resolution is limited by the cell size and not by the process, unlike in 3D printing. Let me just get rid of that. So as I said, in 3D printing, you build these structures, as you can see, like these red arches that we build, but you also have to build this blue structure which supports it. In electrospraying, you don't have to build that blue structure. And that means time is reduced for finally printing a 3-dimensional structure and an overhang that does not require supports. These what a 3D printing bone architecture looks like. You can see that the support holds the part of the bone during its printing stages. You can also see here, I'm showing you articles that have been published by independent groups showing that 3D printing damages proteins during the printing process. You can see here that cells are also being damaged due to shearing. And note in this paper and many, many others, those of you who want to search this can do a Science Direct search, and or if not, contact me and I can send you the articles. But there are hundreds of articles showing that only single type of cells have been printed, yet single cells have been damaged. So the idea is to use large bore needles, therefore reducing the shear, but also having the ability to form residues once deposited in the micro or nanometer scale. The ability to use large bore needles will also automatically allow us to put in more cells through that. So these what I think everybody wants. This is what my interpretation of what Americans want. You want to be able to go up to a machine and just press a heart, kidney or liver. Unfortunately, this is as far as I think it will go, as much as a cartoon. But anyway, so these are the sort of tests we have done. I have to say that we've done RNA sequencing as well added to this. But these are the steps of assessment of cells we have carried out. Note that some of these tests are biomedical laboratory-based testing, but we have also used clinical readouts, which is most important if you are actually trying to take something into the clinic. So let me introduce you to electrosprays and electrospinning. These are technologies that have been around for yonks. As you can see, electrospinning has been around by Cooley and Morton in the 1900s. Electrosprays have been first talked about in the 1400s by William Gilbert in the DeBaknet. The significant papers that have come out from this have been the article in Science by Fenn et al. In 1983 that actually showed that electrosprays can be combined with mass spectrometry in order to identify molecules by their charge to mass ratio. What you see, the first image that you see right on top with the rainbow colors is a chamfered needle having a liquid cone which emanates a jet from its apex and then breaks into a myriad of three-dimensional conical droplets, which you see as like a rainbow colored droplets. The message that you've got to take from this picture is you can see that the diameter or the inner bow of the needle, and you can see the diameter of the jet. Unlike in 3D printing, any 3D printing, the jet in 3D printing is double the size or more than the needle. In electrosprays and in electrospinning, the actual jet that emanates is maybe 1/100th the size of the inner bow diameter. Therefore, resolution is not a problem. Electrospinning is a sister technology to electrospraying, and the only difference being the rheological properties. If you vary the liquid from something like something as low viscosity as water, not that high conductivity, or let's say ethanol, and you move to something like glycerol, you will be able to see that you can pull out a continuous filament. That filament on collection forms a random scaffold, and that is what electrospinning is. I've pinched a couple of more pictures from my colleagues in Spain. As you can see on the left page, images on the bottom left, you can see how you can scale up these technologies. The residues, you can see what you can build. You can build some beautiful little drug delivery beads or scaffolds which are porous or solid, and the scale up of this is not a problem at all. And the beauty of the scaffolds is that you can control not only the cross stitching, but also the random nature to some extent in order to make very, very strong materials, which in comparison to other approaches, if you like the spinning, rotational spinning fiber generation approaches, which we see in fun fairs for sugar candy, for candy floss, which are only unidirectional. So there is a strength element that negates the use of that in tissue engineering. So you can see here, I've shown different needle configuration. So in electrospraying or electrospinning, you can have 2-3 needles that are centrally within each other, thus forming structures like core structures or multiple multi-core structures. You can see also at the bottom left-hand corner, you can see a controlled electrospray jet that is stable and breaking into a fine bunch of droplets. You can see when you increase the applied voltage, how the jet starts to whip. And then the final image, the third image, you can see electrospinning, the continuous forming of fibers. There are various, these are the arrangements in my lab. And you can see that we actually spray them inside a laminar flow hood. We also, I should also give a shout out to a company that greatly supports me in this endeavor, which is Bioengineering, based in Switzerland. They give us some wonderful equipment, and we have been collaborating them for the last 15 to 20 years. So the image I told you, William Gilbert published his book in the 1600 about, he was the first one to start the electrospraying endeavor, and you can see the two patents on electrospinning by Cooley and Morton here. So electrospinning and electrospraying have not been, are not new technologies. During my PhD, we did some work where we were able to 3D print architectures. As you can see, these prints are much like 3D printing, where you can see the layering of each layer. But note, they are layer by layer and they are only walls, not overhangs. In 2006, when I was when I had my own independent group, we partnered up with Alice Sullivan, who was at Queen Mary at the time, where I was as well. And we were able to show self-standing structures. As you can see on the top left-hand side of your image, you can see the sequence of image showing the growing of a structure. Almost in the middle, you can see a structure like a bridge that we've grown. Now, I know that people will say that this structure doesn't look like a bridge at all. It just looks like something that has been spat through. My answer to that was, this was the first example of it in 2006, and a baby has to crawl before it starts to walk. Regardless of that, 6 years later, a group in ETH Zurich took our work on and published the ability to make these fine pillars, as you can see, which is a far right-hand corner image. And you can see the aspect ratio of that pillar. Since then, their similar work has shown, they have bettered our work, which is absolutely wonderful to see. And you can see these beautiful pillars. They are all built without supports, hence reducing time and money on the material usage. So 3D printing, in my view, the classical 3D printing, in my view, is obsolete. I mean, I can show you more pictures about that. I didn't want to waste time on that. But if you're once again interested, please come to me. I'm more than happy to show you. Now, with making a scaffold for tissue engineering, what a lot of people do is they make a piece of gauze-like structure and they seed them with cells and they transplant it. The problem with that is that the gauze porosity determines the full integration of the cells. And added to that, if you layer a bunch of cells on the first layer of gauze, if the piece of gauze or scaffold is thick, the next bunch of cells that you deposit on it through manual or automated deposition is not going to go through, because once cell deposit on these scaffolds, they spread. Once they spread, they start blocking the actual pores. So infiltration is a problem, and various other applications, as you can, issues here. And electrospun scaffolds have been used as condoms. A colleague of mine in the US has used them and shown that. And that shows that these scaffolds electrospun form very small pores, and therefore even the smallest self-propelling cell can't go through. If that is the point, if that is an issue, then how can a adherent cell do that? You can see here her articles. She's got many, many articles that she's published. And these are one of her patents that it's Kim Woodrow that you should be looking up. We also showed, this was some of our work, we also showed that you can punch the back of a mouse. Obviously, that's not new, but we made a scaffold with PLGA and various other materials. And you can see over a function of time, the actual scaffold alone does not allow the wound to completely heal. Obviously, the mouse wounds heal in a different way from the outer to the inner, as opposed to how our scabs form. But nonetheless, it goes to show that thick wounds do not heal as well as they should. In fact, you can see here the column of images on the extreme right. You can see where the square is shown, the topmost layer on the skin starts to curl upwards, therefore not being able to go in because the pore sizes are so small. And therefore, you encounter a problem which we refer to as foreign body reaction and rejection. This is the article that we published. David Becker is a very well-known scientist in wound healing. He's unfortunately left UCL to Singapore, but we've done lots of work with him and shown that just scaffolds are not good enough. Now, the idea of taking bioelectrosprays and cell electrospinning to the tissue engineering endeavor is, I told you that we take a biopsy from a patient, grow the cells outside the body, grow them in individual populations, and whatever population that doesn't grow, we bring in induced pluripotent stem cells. What we then do is take all those cells, mix it with a polymer, and build a tissue. And that's where we come into it. And this is just an image to show you the schematics of it. But this was the first paper that we published in 2004 that we showed that electrospraying can be used for spraying cells without killing them. And you can see here we did laser spectroscopy on the right image. You can see it's a trimodal distribution. Obviously, the two peaks are the ones that contain cells. The others are just blank. Nonetheless, this article, which was published in Small, showed that you can process cells without killing them. And I'll explain to you why we use, in electrosprays, we use a voltage of about 10,000 to 20,000 volts. And why cells don't die. I'll explain that to you next. You can see these cells, these are Jurkat cells that they are, after spraying, they're undergoing mitosis. We increase the applied voltage even more, and you can see sparks are flying between the ground electrode and the needle, and yet the cells are not dying. The image on your right shows the liquid that we have labeled and the cells that we have labeled within the encapsulations. So we've, at the moment, we've done nearly 600 different human cell types, including a wide range of stem cells and even whole organisms, to which I'll show you some articles as well. The reason why cells don't die is that our current is very low. Unlike electroporation. Electroporation is a technology that's used by geneticists, and what they do is they make the membrane of the cells leaky by increasing the applied current, and thereby getting your construct into the cell. During the process, sometimes the cells repair this membrane leakiness, and sometimes they don't. It's a problem with the technology, and geneticists have learned to live with it. There are, at the moment, from what my experience with electroporation, that the studies we have done, we've seen a cell death rate of about 60 to about 70%, depending on cell sensitivity and cell type. So the difference between our technology and electrospraying, although our voltage is very high in the thousands of volts, our current is in the nanoamperes, about 5 to 10 nanoamperes. Now, taking this to the clinic, if there is prior art, it's obviously much easier. Electrosurgery or electrocoating is a technology that is highly used in the clinic. And this is for cutting various, molds and various other things, or even cutting tissues. And what you're using is you're using a tissue spark to cut tissues. So this is the idea why tasers don't kill, taser guns. The voltage is high, but the current is low. Obviously, unfortunately, some people have died who have cardiac problems or issues. Nonetheless, it's the current that kills and not the voltage. You can see here people have been using electric fields to kill cancerous cells. They've been used to stunt fish for vaccinating and all of this sort of thing. So the problem that I showed you before is, although we are able to spray the cells, because the actual cell solution has a high conductivity, the cells spray in unstable mode. So we went back to the literature, and what we were able to do is to find that you can put one needle within another, coaxial needle. You can see that the inner needle has a red liquid. Obviously that's a cell media that is high in conductivity and low in viscosity, and the outer needle holds a liquid, a polymer, that is high in viscosity and low in conductivity. And therefore we were able to get a mono distribution of droplets, or a mono peak. This was the article that we published in 2006, showing that you can actually make stable jets from very high concentrated cell solutions. All our cellular studies have been carried out using flow cytometry and well-known established protocols. We have, as I said, we've done about 600 human different cell types and primary cell types. So there are a whole host of papers that we have done. These are some of the papers that we have published on primary cardiac cells and various other cell types. And we've made patches for hearts and various other functions. Added to that, you can see that we have also taken, the beautiful thing about being in UCL is that we have so many wonderful biologists. And one of the biologists that I happened to go to collaborate was a gentleman who works with zebrafish. So we took zebrafish at a fertilized egg stage, and we sprayed it, and we looked at their development. And you can see the two images on the left, their brain fold and their brain development are unperturbed. So the electric field, or as I said, the voltage has no harm on the effect. And why would you do this on whole organisms? Well, we want to be able to see whether if we sprayed it, this is not done by us, but you can see there's a dog here with a tail growing from its head, or a cat with two heads. We want to see whether we will perturb the embryological development. And our studies show that it didn't. So these are all the organisms we have sprayed with before: zebrafish, Dictyostelium, Xenopus tropicalis, the frog, African long-called frog, C. elegans, which is the worm, and Drosophila, the fruit fly. All of these organisms we have sprayed using our technologies, both bioelectrospraying and cell electrospinning, and shown that they are not perturbed. Obviously, the government in the UK, the Home Office, didn't allow us to take the zebrafish or the C. elegans and, sorry, the fruit fly and the frogs to over a certain stage. But with regards to worms and Dictyostelium, the Home Office doesn't care, and we took them all the way. And we showed that their ability to reproduce is not perturbed or anything like that. These are some of the articles that we have published. So you can see that since our publications, we showed that you can combine not only the ability. Now earlier I said John Fenn was able to identify molecules by electrospraying, but now you can electrospray cells and identify cells and various cell surface molecules. And that has been recently carried out by a group in China, I believe. And they have been able to analyze single cells super precisely. You can see other people have taken on our technology and been able to build three-dimensional structures in 3D. And you can see, I can show you, this is where we actually took electrospinning and we put cells into it. And you can see the fiber with the cells inside. This was a paper in 2006, the first example of it. And these are the sort of tissues that you can build, three-dimensional tissues. We were able to submerge the needle as well. And that is for obviously trying to assist spider bifida. You can see here where sometimes children are actually taken out well before their date of birth. It's called second birth. And you actually repair their infarct neuronal stem that doesn't close up, and then repair it and then put it back into the womb. But we are now able to inject a needle that is able to actually spray stem cells from amniotic stem cells into the infarct area and try and press that down. That is a collaboration we are doing once again in UCL, and the article will be soon out in a journal. We've also done human sperm. We've shown that you can encapsulate them and actually freeze them down. Is a better freezing method where we can actually recover a higher population of viable sperm cells, which is very important, not only for human use, but also for agriculture and aquaculture. You can see the beads that we have incorporated the sperm cells into. So there's a whole host of things you can do. Now, these are vessels that we have built as well. We've incorporated genetics into it. So not only you can take your own cells and genetically alter them and introduce them back into the body, meaning your cells are providing the therapy for you, therefore there's no rejection. And by making these beads, you can also make models. These are another article that we published with respiratory consultant Paul Elkington in Southampton. He's a guru with regards to respiratory medicine, and what we showed here is that we can actually see, we can actually model human tuberculosis. Several papers that we published with that group, and we are now making various other 3D models as well. Here, once again, you can see that we have made these beads that are vascularizing, and what we've done is we've collaborated with colleagues at King's College, and we've inserted that into mice. And we've been able to revascularize the issues with vascularization issues in legs of mice. Now, the whole point of this is that once, sometimes when we grow old, we have issues with regards to vascularization in our legs. And therefore, hopefully we will be able to one day take this into the clinic for human usage. These are some studies that we've done that we can insert the sprayed cells into the tail vein of the mouse and see whether it homes, its capacity to home back to the lymph nodes. And we have shown, we have done quite a lot of viability studies, because if the cells are not viable, there's no point looking further. We've also built various other tissues, skin tissues, looked at various aspects, cell division, viability and functionality, and inserted them into mice, as you can see here. Followed the cell migration over a function of time. And we've obviously done the same work for both technologies. So these approaches are becoming platform technologies, not only for building tissues for repair, replacement, and rejuvenation, but we can also build a tissue in a dish to start testing various drugs, and also to freeze down cells and to store them, to expand them in a scaffold, which are in 3D, unlike in normal cell flasks. So the applications are quite wide-spanning. These are the vessels that we have built, which have been put into mice, which have been able to show that they are rigid, and they rigid as much as close as possible to native tissues. And the whole point is, now everybody, all our competitors' idea, and I stress that, I don't say our competitors, but I say that the competing ideas, because this is what we are doing. We are challenging competing ideas. Now everybody makes a scaffold, which is the top image, makes a scaffold, seeds the cells, go into the bioreactor, and then comes back to seed the cells, and that's a repeating process till it actually fully feeds into it and then goes into a human. We are able to make scaffolds and whatever structure with cells inside, because our cells are in the fibers. Therefore, the bioreactor time is reduced, and we can go into the human much faster. As I said, you can build three-dimensional scaffolds and actually culture cells in true 3D rather than in 2D. Rather than cell pelleting, we can actually put cells into fibers and freeze them much quicker. And you can control the freezing process as well, because you can have 2, 3, 4 layers on these fibers. So you can control the way you freeze the cells, freeze down the cells, and then thaw the cells. So the whole point is, when you make these scaffolds, you can build a whole variety of scaffolds with our humans, with the patient's cells, and then you can decellularize them and keep them in a tissue bank for recellularizing when required. So the whole process is being turned upside down, and we particularly think that we'll be able to contribute a great deal. In fact, we are currently working with the Department of Defense in the US, with Pfizer and various other companies, and they are funding this work. There are various other approaches that we have taken it to make stem cell combinatorial technologies. You can see here, I'm not going to show you much, talk to you more about that, but you can see the sort of applications you can carry out with these approaches. And finally, these are the sort of images when we see that we want to be able to contribute a great deal and alleviate human suffering if we can. So these are things that keep us ticking. So these are the publications we have done for various journals, and there are many more coming out. And these are the guys I thank a great deal for paying for our research and letting us carry out our research in my lab. Thank you so much. Thank you so much.
Thank you very much.
Do you have any questions?
Suwan Jayasinghe.
I think I hope I've kept the time.
It's a wonderful. Yeah, no, yes, definitely. It's so wonderful. I didn't even feel that time is passing. And thank you very much for, enlightening us. And also now I can see you have kind of spoken about. Hello?
We can hear. Yeah, we can hear. Yes.
I can hear you. And it's so amazing to see how much you can do through this.
I can't hear anybody.
Yes, hello?
We can hear you, Professor Suwan, and I can hear Professor Rohini.
Yes, Professor Suwan, can you hear me? Professor Suwan, can you hear me? I don't think you can hear me. Professor Suwan, I need your response.
Professor Suwan can't hear.
Suwan, I can't hear you.
Wait, I will call him. Suwan, can you hear us? See whether you are muted.
Hello? Hello? Professor Suwan, we can't hear you.
Hello?
Hello? Hello?
I can hear you, Suwan.
Can you hear us?
Yeah.
Madam, we can hear you and Professor Suwan also. I don't know why.
My Professor
Suwan one way, no?
Yes. Yes, we can hear you as well as Professor Suwan, but not he can hear you, I think.
I know he's not responding.
Because he cannot hear you, something is there, no? I don't know whether he can see the chat box. Why don't you drop something in the chat box, Nali, Rohini?
We have, madam, we have.
Yes.
Lahiru, do you know what is the reason?
I'm not sure, Dr. Gayatri. In fact, I think this is what happened earlier as well. We could hear each other, but Professor Suwan couldn't hear us. So
Yes,
let's see.
Yeah. At the beginning also, it was like
Exactly.
I'm on the telephone to you.
Yes.
Okay, Professor
Suwan is on phone with me. Shall I connect through this phone? Yeah. Let me see.
Yes, please. Yes, please. Hello? Hello, Professor Suwan.
Suwan, can you hear me now?
I can hear you, darling.
Okay.
This is through my WhatsApp phone now.
Okay. Nalin, you heard my presentation, yeah?
Yes.
The presentation was perfect. There's no question about it. Yeah. We heard everything, but you can't hear us now.
Yeah, I can't hear you guys.
Okay, so can you hear me? Can you see me at least?
Hello?
Hello, can you see me, Professor Suwan, through the...
Madam, I think he can see you, but he can't probably hear you. He's only hearing what Professor Nalin is saying through the phone.
Hello, Professor Suwan.
Hello, Rohini. How are you?
Oh, wonderful. Thank you so much. We are very thrilled to have you here with us for our first webinar series, and it's amazing that how you presented your thing, on and how much work we can do to live cell electrospinning, and not only 3 R's, that means repair, rejuvenation, and restart. You were talking about some other amazing things that you can do to cell electrospinning. So now it's the question and answer time, and this particular thing will be moderated by Dr. Lahiru Vijayaratna. Dr. Lahiru Vijayanayaka. So how do you kind of hear us, because our students they like to kind of have some questions and to share some questions and answers from you.
I mean, on the screen, all I can see is everything is muted.
Oh, really? So the Lahiru Vijayanayaka, Dr. Lahiru Vijayanayaka is the Vice President for the SLAAS section E2. So now over to you, Lahiru. Can you just moderate the question and answer session?
Yes, madam. Thank you very much. Madam, I'm here, but there's a small problem. I wonder if Professor Suwan could hear me at all. So there might be a problem communicating with Professor Suwan Jayasinghe.
I can hear him through the phone, but I can't hear him through the computer.
It's okay. Now, Lahiru, go ahead. I have put my phone next to the speaker.
Okay, thank you very much, Professor Nari. So once again, thank you very much, Professor Suwan. So can you hear me, Professor Suwan?
Yeah.
Okay, so once again, thank you very much for that very nice presentation. It was very comprehensive and very clear, and indeed very interesting as well, sir. So like Professor Rohini mentioned, now it's time for the questions. So we would like to open the floor for the discussion. If there's anyone who's willing to ask any questions from the speaker, please let us know. And I would like to remind you, you can unmute yourself and raise your question, or else you can raise your hand and we'll prompt you for the question. Or instead, if you are more comfortable, you can put the question in the chat box as well. So to start off, is there anyone who has a question?
Lahiru, there's a question in the chat.
Yes, madam. So I will directly go out to that question. Sir, Professor Suwan, the question is actually from one of the participants. The question is it possible to regenerate the human brain? Is there any approaches that? Yes, sir.
Well, you can regenerate the human tissue of the brain, but whether the functionality is something that we will need to find out how to test.
Okay, sir. So does that mean in the same manner that you explained for the other organs and the other materials that you have developed, you can do the same sort of thing for the brain cells as well?
Yes.
Correct? Okay.
Absolutely. Sir. It is in our opinion that we can regenerate any tissue, but one has to be very careful in order to analyze it to assess whether it actually is a functional tissue.
Okay. So thank you very much, sir. That's one of the questions that was in the chat box. And once again, regarding the technique that you mentioned, sir. So you mentioned that the cells does not undergo any damage during the electrospraying because of the very low current. So just out of curiosity, is there any localized heating that is generated during the process?
No, because it is all carried out in ambient conditions, and generally there is no heat that is generated because of the needles are sort of not extremely sharp at the edges.
Okay, so that means that neither the voltage, the current, nor the thermal energy would be distracting the cells. That's very nice to hear.
Yes,
sir.
yes.
And at the same time, sir, another question that I had was, in the technique that you mentioned, how do you control the three-dimensional morphology of the the structure that you develop? What is the driving force?
Well, you can, once again, you can build it by layer because we are using large bore needles, we can process high viscosities.
Okay.
So because of
Okay,
that, you can actually once the deposit, once the cells are deposited, the integrity is maintained.
Okay, sir.
Also, you can cross-link it, cross-link the cells and the hydrogel by either UV or temperature dependent, or there are various approaches.
Okay. Thank you, sir. Thank you very much. And one question is another question is about the biocompatibility of the organs or the cells that you develop in this method. So based on the materials that you incorporate into the matrix, is there any studies already done in terms of understanding the biocompatibility of the materials, sir?
Well, the biocompatibility issues that we assess are, for example, the polymers, obviously the cells are from the patient itself.
Yes, sir.
And the polymer that we use are inert polymers.
So that minimizes the chances of any biocompatibility issues.
So that addresses all biocompatibility issues.
Thank you, sir. And so there's one more question in the chat box that's raised by one of the participants. This is regarding organ rejection. So the question is, organ rejection is one of the major problems with organ or tissue transplant. How about the rejection and immune response to the electrospray tissues? So I believe that's you answered the question partly. Is there any more thing to add, sir?
We don't see any rejection at all, because obviously we are using the patient's own cells, and the material, the polymers that we are using are already in the clinic.
Okay, sir.
So there is no rejection at all.
Okay, thank you, Professor Suwan. And
no problem.
And the questions keep popping up, and there's a few more questions that has popped up just now, sir. There's a question about using this technology under any microgravitational environment. Can you add any comments on that, sir? Can we use this new? The question is, can we use this new technology under any microgravitational environment?
Yes, you can. I mean, for example, you're talking about using it in space and so on.
Yes, I believe microgravity is a space, I think. Yeah.
Yes, we can do that. That's not a problem as well.
Okay, sir. Thank you, sir. And then one more question that just came up. What are the solvents that can be used for electrospinning cells? Is it only water, or are there any other solvents that would facilitate fast spinnings?
Well, that's a very good question, because solvents are very important. So what we use is either cell media or water-based gels.
So it's mainly water-based.
Mainly hydrogels, yes,
okay.
Water-based.
Okay, thank you, sir. And then we also spoke about the low ability to damage the cells using this technique. There's a question about the ability to damage DNA and the gene expression levels. So is there any chance of damage in the DNA?
No, because
yes, sir, please go ahead.
The DNA, we have looked at DNA and gene expression, and we see that the gene expression is indistinguishable with those gene expression of control cells.
Okay, sir.
So so genetically we are not changing it, nor are we changing it phenotypically.
So there's no chance of DNA damage during the using this technique.
No, not at all, and that is because the current is very, very low.
Okay, sir. Okay, that makes perfect sense, sir. So those are all the questions that were raised by the participants in the chat option. So thank you very much for your comprehensive explanation in each case, sir. Are there any other questions? Is there anyone in the audience who would like to comment or raise any other concerns?
Hello, sir. I have a question. I'm Kaushalya Pereira. I just want to know whether the nerve cells can also be regenerated. And another question, what about the legal background? Like, do you have to take special permission from any legal authority to do all these regenerations and all?
Firstly, the question is, we have to get permission from Home Office before we do any of this work. We have to get special permission to do all this work because we are human cells or even animal cells. We have to have permission. With regards to neurons, we have been able to spray neurons, but we haven't looked at, we have been able to spray neurons and find that they are undergoing cell division and all the cellular behavior. So with regards to regeneration, we can regenerate them, but we haven't built a scaffold for the brain, if you like.
Lyro? Lyro, can you hear me? Lyro? You are muted. Lyro, you are muted.
Oh.
Lyro is muted, Gayatri.
Let me see. Lahiru. Okay.
1 second.
Yeah. Now you can talk, right, Lahiru?
Okay, Dr. Gayatri. Yes. For a moment, I couldn't unmute myself. Now it's okay. Thank you very much, Dr. Gayatri. So thank you, Kaushal, for that question. Are there any other questions from the participants?
Yes, Lyro, I have a question.
Please go ahead, Prasanali.
Suwan?
Yeah.
You are a mechanical engineer, okay?
Yes.
And what made you to inspire to go into the biomedical and this hardcore biomedical engineering? And can you inspire our younger generation with the mechanical engineer typically is mechanical engineers are not working in this field much. So can you inspire the younger generation? We have about 75 to 80 people over there.
All I can say is to everybody who's listening as students, all you need is a curious mind. In my case, my father was ill, and that's what inspired me to pursue something in biology. And I don't mean to upset any engineers, mechanical engineers, but wanting to go in a car that goes 0 to 60 in 4 seconds does not excite me anymore.
Okay, that sounds good. That sounds extremely exciting. And I have another question, Suwan. There are some UCL scientists working on gyro spinning.
Yes.
And your one is the electrospray, and we are also working on electrospinning with various other applications, not tissue engineering as such. But I think you explained the difference between the gyro. You did mention the gyro spinning, but there are problems associated with gyro spinning in terms of the strength of the material. Can you elaborate on that?
The thing is, when you build a structure that is fiber based, you want to have cross stitched. If you don't have cross stitching, for example, if you wear a shirt which has fibers in only one direction, the threads, the shirt will fall apart.
Okay.
So you have to have it cross stitched. And another thing is, with regards to using this technology of rotary spinning, you can't, some of my colleagues in Brazil tried to do this, and they found that the cell entrapment is not successful.
Okay.
Within the fiber, because of the centrifugal force is so strong, the cells spin out because of their higher density.
Okay, got it. Yeah, because that is mainly the centrifugal force. You think that's unidirectional?
Yes, yes.
Okay, all right, got it. Yeah. Thank you very much, Suwan.
No problem. It's a pleasure.
Yes, now it's time to Dr. Gayatri.
I think the questions are over. I think Rohini will take over.
No, yes, okay. Dr. Gayatri. Hello, Prof Suwan.
Hello.
Officially, the official vote of thanks will be given by Dr. Gayatri, Secretary of the SLAAS. So over to you, Gayatri.
Okay, thanks, madam. So on behalf of SLAAS E2 2021 team, I would like to thank Professor Suwan Jayasinghe for accepting our invitation with a very short notice and share your expertise and most up-to-date scientific information on how we can use this novel non-destructive biospray approach to process living cells, which can be used as therapeutics and to build tissues and perhaps complete functional organs in very near future. So we wish you all the very best to drive your research to the next dimension in this new year, 2021. And we are eagerly waiting for the exciting scientific developments in this area coming from your lab. So thanks again for honoring us with your presence here today. And Professor Rohini, I think we can wind up for today. And thanks everyone for your participation.
Well, before you say bye, let me say thank you very much. Let me say thank you very much, Dr. Gayatri, for having me and hosting me. And I also like to thank Professor Rohini De Silva for inviting me.
Okay, sir. Thank you so much, sir. It's wonderful and so nice to have, and it's a pleasure. And we had a wonderful session with you. And thank you so much. And as Gayatri said, wish you all the best in year 2021. Thank you, sir. A round of applause.
Thank you. Thank you.
A round of applause to you.
Thank you.
Thank you very much, Suwan.
Thank you. Take care. Yep. Bye-bye.
Thank you.
Pleasure to have you.
Bye.
Bye-bye.
Okay, bye-bye.
Bye.
Bye-bye.